Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Viruses. Don't you go giving them a bad rap. Viruses. It's not that they just do the bad crap. Viruses have the potential to do good things. Viruses. They can control fungal pathogens. Bad rap.
Wow, Robin, what a wonderful rendition. That's an expert rap by an expert virologist. Welcome, everybody, to another Sigest podcast. I'm Hilary Ireland, and I think that's the best welcome we've ever had onto Sigest. Welcome, Robin. Welcome to the podcast.
Thanks, Hilary. It's great to be here.
So in today's sometimes frightening world of COVID-19, one would be forgiven to thinking that viruses are all bad and to be avoided at all costs. I'm Hilary Ireland, and today we're speaking with Dr Robin MacDiarmid, a virologist at Plant and Food Research, who's come to tell us that these often misunderstood particles are not all bad and may even be very beneficial in some cases.
I hope to give you some insights around this. They don't all have the bad rap, but we've got a lot more research to do to understand that.
Right, cool. So before we hear all about your defence of viruses, can you tell us a bit about yourself and your role here at Plant and Food Research?
Sure thing. So I'm a principal scientist at Plant and Food Research. Here we are in Mount Albert, the headquarters. However, I do live up in Kirikiri in the Bay of Islands, so I'm doing the commuter bit between those. I lead the viruses and like organisms team, but I also work at the University of Auckland, so we have quite a few students here, masters, PhD, etc., based at Mount Albert, where they're doing their thesis research.
And you're a supervisor of
So I
those
supervise.
students, right,
So I
okay.
supervise those students along with some fantastic co-supervisors. Yeah.
Great, cool. So what drew you to the study of viruses in particular?
Yeah. So that is a bit of a story. I grew up Kirikiri, where I now live. I grew up on an orchard, and citrus, subtropical. Subtropical means at that stage anyway that we grew tamarillos. And I was very aware of my father being frustrated with this virus that was infecting tamarillos, causing reduced yield and the blemishes that you see, those dark blemishes on the fruit of tamarillos. Mm-hmm. And he was always frustrated about them. So as I went through my bachelor's and master's, I was finding out about these things, and I realised that we could approach the management of this using a number of different methods. So I launched myself into my PhD studying that very virus in tamarillos.
Cool. This isn't a Science Life for Me podcast, but I'm interested to know, did you study ecology and biology in your undergrad
interesting.
years?
Yeah, so in my bachelor's I studied biochemistry. I took no botany and I took no microbiology. And then for my master's, I studied deer antler development. You can see the disconnect. And then I was influenced by a marvellous speaker, Gail Timmerman-Vaughan, who spoke about a related virus, potato virus Y, and what she was doing in potatoes. Mm-hmm. And I knew that potatoes and tamarillos were related. I didn't know whether potato virus Y and this tamarillo mosaic virus were related. I went immediately over to the science library and found out they too were related. Chatted with Gail Timmerman-Vaughan, and that was really launched me into my PhD. So that's when I actually came up to Auckland from Dunedin, where I'd been doing my bachelor's and master's, and did my PhD with Richard Forster and Richard Gardner at the University of Auckland. So I'm sort of phenocopying that relationship now, because I'm based at Mount Albert supervising PhD students and also the supervisor through the University of Auckland.
Cool, full circle. That's wonderful. So people probably know more about viruses these days than they ever have before. But for the sake of clarity, could you tell us, what are viruses exactly? What are virus-like particles?
Yeah, so viruses are actually one of those organisms. We call them organisms, although they're not actually alive. They're totally dependent on their host to replicate, but yet we call them an organism just for ease, and they are not well defined. So you can find different formal definitions of them around the place. But essentially, they're made of a nucleic acid, so that could be either DNA or RNA, and they depend on their host cell for replication, although they do contribute proteins towards controlling that replication. So they do encode proteins, and that sets them aside from other virus-like organisms, such as viroids. So viroids are only RNA, and they don't encode any proteins. So they are very, very funky and really effective. So they're totally dependent on their host. They're actually very stable and can really control their host just through their RNA that encodes no proteins. Viruses do encode proteins, at least one, sometimes many. And all organisms are infected with at least one virus. They're a host of at least one virus. And some of those viruses are tiny, just 2,000 bases long. Some of them are huge, nearly the size of a bacterium.
That's incredible to know about the different size differences. I had always imagined they were more smaller than they were larger, but fascinating.
Yeah.
You talk about virus-like particles just being made of RNA, and protein coats and things provide a bit of stability in external, say, environment. Are those RNA-based virus-like particles, are they just kept within the plant and transferred from plant to plant, like through insects or something? Or
so
how do they protect themselves?
That virus-like particle, or it is a virus, it's a virion. And so that will be composed of a nucleic acid, so either RNA or DNA. Might have one strand or multiple strands, and it might be packaged in one container, or it might have several containers.
Okay, complex.
Yeah, there's a lot of variety in virology. And they have to get from one host where they're replicating and making that virion, those containers, to another host somehow. So we've seen the pictures of SARS-CoV-2, the virus that causes COVID-19, and we are all very familiar of it. It's this round ball with spiky spikes coming out of it, and that's to get from human to human. So plant viruses, which I'll speak mainly about but not exclusively, are often transported by sap-sucking insects. So on the phloem goes the mealybug or the aphid. Up it draws the virus, and then or as it sucks on the next plant, and that allows the virus to go back into the new plant host and then replicate.
Got it.
Now, that's only one way that a virus might be transmitted. Depending on the virus, it might be really, really stable, such that just touching that plant, and then the infected one then touching the uninfected plant, might be sufficient to transmit that virus.
Amazing.
Or the virus might kill the plant and then get down into the groundwater, and then move through the groundwater and get into the next plant through a damaged root. So the relationship of the virus with its host, here we're talking about virus and plant hosts, sets up, it's evolved to best get transmitted. So yes, the virus that kills the host does so to get down into the groundwater and then get to infect and replicate in the new host. But a virus that is transmitted by an aphid doesn't want to kill the host, because an aphid's not going to suck on a dead plant. So the virus that's, say, transmitted by an aphid will actually causes the leaf to change to attract the aphid.
Amazing.
Yeah, this is just a mind-blowing, phenomenal world of utter control of viruses. Get totally used to the fact that viruses control the world.
That sounds kind of scary. Actually, that kind of plays into the scariness around COVID in a little way. But have you found the wider understanding of the viral terminology, thanks to COVID, has been helpful to your work? Yeah,
it's been great. I mean, look, I know that COVID-19 has been very harsh, particularly outside New Zealand. And at the time of this recording, India is going through an explosion of virus incidents, COVID-19 disease incidents, and it's a really dreadful situation. Everybody in the world knows about viruses now. And the language, shared language that we all understand in terms of quarantine, in terms of testing, the process of the test of the real-time PCR is getting understood. Managed isolation, so that the language is that everybody is aware of, familiar with, has deepened the general understanding of virology. And that means when I go and talk with somebody in industry, well, we now have this raised bar of communication. We can just like it to COVID-19 and boom, we're just understanding each other very, very easily. It's made life a lot easier for me. And I think that we can really benefit in, say, horticulture from that increased understanding.
Got it. But with, for instance, like managed isolation of plants, would that happen, or would you just destroy them?
Yes, we're not quite so considerate of plants, virus-infected plants, as we are of virus-infected people. Often the way to manage, say, an orchard or a vineyard is to identify those virus-infected plants and then to pull them out, destroy them. That's called a process of roguing. Although, if your plant is very valuable, you might want to make the effort to remove those harmful viruses. And I want to distinguish that, the harmful viruses.
Yes.
So that you can produce plants that don't have those harmful viruses. Now, a lot of our horticultural plants that we grow in New Zealand are grafted. So we go actually through quite a lot of care. I'll use the grafted grapevine standard that the viticultural sector uses. We go through quite a lot of care to remove harmful virus, a virus called leaf roll virus, from the nursery stock so that we're planting healthy plants in the vineyards that don't have this detrimental virus. So we do go through a sort of managed isolation process where we're testing and making sure that that mother stock from which we're grafting is free of that harmful virus. And we do test to make sure that before they exit that managed isolation, that they are not carrying that virus.
Right.
Yeah.
Just for my understanding, when you say remove the harmful viruses, is this actually removing the harmful virus from a plant, or is this removing the plant with harmful viruses?
Yeah. So you can do both of those.
You can.
So
Okay.
you know, if you then find a plant in the orchard or in the vineyard with that harmful virus, then the best way is to actually just directly remove that plant so that it's not going to infect others. It's creating a reservoir. But if you've got a valuable plant that has a harmful virus, you can go through some treatment protocols to remove that virus. So
Very nice.
viruses are transported throughout the plant through the phloem, and the meristem at the very tippy top of the growing points doesn't have a strong phloem connection. So if you cut that very meristem tip and propagate the plant from that,
Yes.
then you're less likely to have the virus.
Okay, cool.
Likewise, you can treat the plant with heat or cold or chemotherapy, and that knocks the virus down, and then again take that meristem tip culture and just reduce your chances of propagating the virus, and then boom, that one there is the one that's virus free, and so you propagate from that.
Cool.
Yeah.
Understood.
Yeah.
Great. Thanks for clarifying that. And here we are. We found ourselves partway through this podcast talking a lot about harmful viruses. So we've fallen into the trap already. So are they all bad?
Yes. Well, why did I say yes? No. No. We have So viruses, I forgot to say, are very small. They're smaller than the wavelength of light. Okay, so you can't see a virus. You can see the symptom of a virus. Okay, so right there, you're just looking for the ones that cause a symptom. You're looking for the ones that cause a disease. And this is, so virology has been very technology driven or technology constrained. And of course, initially, we only had the technology of our eyes. And so we saw these symptomatic plants, and then we try to work out, is it a fungus, is it a bacteria, is it an oomycete? No, it's a virus that's causing these symptoms. And so we found that this plant had this virus, and so it caused maybe tobacco. We found it in tobacco mosaic. That's the symptom. It causes virus, tobacco mosaic virus. So we named that all very happily. Off we go. We find tulip breaking virus. So name that. So we found all of these viruses that cause symptoms. And in finding all this collection of viruses that cause symptoms, we induced that all viruses cause symptoms. But we were totally constrained by our technology because we couldn't see those viruses that did not cause symptoms. So how do we see those viruses that do not cause symptoms or that could even be beneficial? That's a lovely plant. Very good. Well done, plant. You're looking very lovely. We think it's the plant, but I would suggest that in many cases it's the virus of the plant that's making it very lovely. And so it wasn't until we got this wonderful technology of high throughput sequencing that we got to start to scratch at the very surface of this whole other world of viruses. What are they doing? So we're finding all of these sequences. We have no idea really about their biology because, trust me, doing the sequencing and finding a new virus is quite satisfying, and it's a lot cheaper than doing the biology of asking the question, what is the impact of this virus?
Yes.
So that's a lot of mahi to do, to work out the impact of a virus. But we're finding viruses associated with positive phenotypes. The virus-infected plant or the virus-infected fungus or whatever other organism can have some advantage. So an example of that is, well, there's many examples of that, and I'd love to run through a few of them with you.
Yeah, please do.
Yeah? Okay.
Yeah.
So, during my PhD, I didn't always water my plants. Sometimes there were better things to do in the weekends, and especially over summer, hot. And I'd come back and to my trays of uninfected controls and my infected plants, and in retrospect, I remember thinking, oh gosh, these virus ones actually look quite perky. Now, somebody actually looked at this phenomenon and studied it and asked the question, do viruses confer drought tolerance? And in some, not all, but in some cases, the answer is yes. So there's a fantastic researcher named Marilyn Rusink. Hi there, Marilyn. I hope you're listening to this. Whose students studied this phenomenon and found that in a range of viruses, these are what we'd normally call pathogenic viruses, they're actually also conferring this beneficial trait. And one virus might do that in a number of different plant hosts, and there are a number of viruses that do it in different plant hosts. So there's quite a mix and match thing going on here. So that really does ask the question, what is the role of viruses in evolution? Can a virus help a plant before the plant has the ability to reproduce, and evolve, that it might take a decade or a century or longer to evolve to that drought condition. But can the virus help that plant in the meantime survive, to reproduce, and then to evolve its best drought coping mechanism?
Wow.
Yeah.
Fascinating. I think you talk about Marilyn Rusnik, and I read one of her studies involved a complex association between not only viruses and plants, but viruses, fungal endophytes and plants.
The menage a trois.
You're right.
Yes.
Can you tell us about that
Let's
story?
just have a little look at that. So, romping through the Yellowstone National Park, skipping away down the path, she and her colleagues saw these plants. It's a grass called panic grass, growing right beside the hot pools, above the temperature of 50 degrees. It's like, whoa,
Wow.
that's,
Unusual.
this is warm on my feet. How is this plant surviving here? So they had a look at these plants and found that they were infected with a fungus. Oh, they say the fungus is helping to confer the heat tolerance. And they found that whenever they saw the grass growing above 50 degrees, it was infected with this fungus, and those ones that didn't have the fungus couldn't grow at 50 degrees. Happy story. But they dug a little bit more.
Mm-hmm.
And they found that not only did that grass require the fungus, but that fungus had to be infected with a virus. So only in the case where the virus was infecting the fungus and the fungus was infecting the plant did the grass grow at 50 degrees or more.
Wow.
And so that's the type of intricate relationship that we're starting to uncover, where there's this very beneficial symbiosis going on
Right.
between three different kingdoms.
Mm.
Yeah,
Yeah,
it's
amazing.
a fascinating world, and the virus is able to pull the strings, the biological strings, like a puppeteer, to its advantage, because of course it wants the fungus to survive,
Yeah.
and to survive the fungus needs the plant to survive.
It really challenges our definition of a living organism, doesn't it? Really, I mean, we have a definition of life, and it, with those 7 characteristics that I can't relate.
Or Mrs.
offhand.
Green. Yes. Yeah.
But many, viruses don't have many of those things, but it's particularly sensing. But you sort of have to wonder if there is some kind of...
Well, look, you talk about sensing. A virus replicating in a plant can sense within milliseconds the landing of an aphid onto the plane of the leaf of the plant.
Wow.
And it goes from one body within the cell and scatters out throughout that cell so that it has more likelihood, somebody in its population has more likelihood of being sucked up by that sap-sucking insect.
Amazing.
Yeah, so can they sense?
Can they sense,
What
yeah.
they do is they tap into the key networks of their host.
It shows a real sort of... It suggests there's a bit of a purpose in terms of how they... But we know that evolution is more... it's random. It's chance events that confer an advantage, and that's how it is
But
propagated.
look at us.
But
Look at us. I mean, here we are plonking along, maybe having a kid at in your 20s or 30s, and maybe one. I'm one of 6. Maybe you have 15 kids. Let's put it out there. 20 kids, one woman.
no, let's not.
Okay, but you get my idea. It's a small
Yep.
number. A virus can replicate to millions within a day.
Right. Yes.
Its rate of replication is just so outpacing its host that it evolves and can fine-tune itself extremely quickly. So the evolution of it and its
Yes.
ability to control its host is outpacing the host by many-fold.
Yeah.
And so no wonder they rule the world.
Yeah.
Did I slip that in?
Yes. Puppet masters ruling the world. It's scary.
Well, is it scary? And
Good point. Yeah, well, so many viruses, retroviruses, they insert themselves into the host's DNA, which really does sound kind of scary in terms of the potential for disruption of the host's genes. But has it always been bad, those events?
Well, we don't know necessarily the outcome of the ones that were bad because they were bad and they didn't propagate.
Sure. So that provides an example for good ones?
Well, and so we're left with the good ones.
Yeah.
And so that whole process of popping into a genome, a high proportion of our genome is viral, provides a change event for which evolution can then select. And so that has enabled things like a placenta to form.
Tell me more. Say more about that.
Well, it's pushing a little bit out of my expertise, but some of the key genes that are required to form a placenta have been activated through the integration of a virus or a virus-like element. Because where does a virus become not a virus? When it's now integrated into its host. It's now regulating the host gene and actually is never getting out of the host. So is it now a virus? So now we're talking about virus-like elements.
Okay.
And so they are forming the promoters. They are viruses, the promoters that are enabling the making of a mRNA copy, a recipe, and then the protein that might go off and do something like make a placenta. Or they might be able to take a gene from one of their hosts and insert it into another of their hosts. We now think of that as GM, but this type of horizontal gene transfer can occur via viruses. It doesn't necessarily occur very often, but when it does occur in a favourable manner, then that event can be selected for.
Yeah.
So,
and it really has. Marsupials, mammals,
yes,
all placenta-based, and monotremes.
They have benefited from this whole process.
Yeah.
Did I mention that they rule the world?
I'm beginning to believe you.
Yeah.
Actually,
Yeah.
I believe you. It's fascinating. Yeah. So, are there other types of benefits that you know of that viruses have brought to their hosts?
So I can go into some examples of so there's drought stress and heat stress and etc. But I'd sort of like to sort of move a little bit more into a benefit that we might think is beneficial.
Great.
Yeah.
Sure, please do.
So for instance, if a virus is infecting something that we think is pathogenic, a fungus that is hard to control out in the orchard, out in the vineyard, then from our perspective anyway, that virus can be considered beneficial.
Yes.
And these are the types of viruses that our team are now studying. They're naturally occurring out there in these phytopathogens, in the pathogens of plants. And from our perspective, they're very useful because they're slowing the growth
of the pathogen.
Of the pathogen.
Yeah, right.
And they're making it no longer a pathogen. They're called hypovirulent, hypo, that's with an O, not an ER, hypovirulent viruses. So they're reducing the virulence of their host, which would normally cause a problem on the crop that we're interested in.
Okay.
So this whole world of viruses in fungi, in phytopathogenic fungi, is opening up.
So how do you begin to find those good viruses that will...
Yeah, so you can go back to your high throughput sequencing and find them. And so new species of viruses are being discovered all the time. New genera of viruses are being discovered all the time. And even new families. So just remembering that, like Homo sapiens, so sapiens is the species, and Homo is the genus, and up above that is the family. And so whole new families of viruses are being discovered that we haven't ever known before because of this process of high throughput sequencing. And then marrying that with the biology that more time-consuming part.
Yeah.
We can find those ones that are associated with this beneficial, from our perspective, outcome. And so you can go through a process of finding these and then asking the question, well, how might we be able to use them?
So is the natural biodiversity available in New Zealand and native forests and things a good place to look for new viruses?
We've really focused on those phytopathogenic problems in our crops. And we're now starting that adventure of looking in our crops to see what viruses are there, and they're there. But oh my gosh, there's this huge world beyond the crops. And you know what? We actually grow our plants, in an ecosystem. What else is out there in the New Zealand, Aotearoa New Zealand ecosystem? We've done a little bit of high throughput sequencing of some indigenous plants that are growing close to horticultural spaces. And every time we sequence, we find new to science viruses. We have no idea what they're doing. No idea what they're doing. So I can't even tell you where we are on that road of discovery, where we are, how much of the iceberg we can see, and how much of the iceberg is underwater or in our forests or even in our horticultural spaces. But we have got a lot to discover. I'd say that we currently know less than 1% of the viruses that exist.
Yeah.
And then, even of the 1% that we do know, we don't know what their impact is. So in terms of virology, it's absolutely a place to be doing research, because there's so little understood about how they are controlling the world.
Right. Yeah, for sure. Just as an estimate, would you say that pathogenic viruses are quite rare then?
Yes, I would. Pathogenic viruses, I would say, are quite rare. And in general, the understanding of virologists at the moment is pathogenic viruses are those ones that haven't yet fully co-evolved with their host, or have co-evolved with their host, and it requires pathogenesis. It requires disease. And the example is that one where it has to kill the plant to get into the groundwater to go to the next plant. But that mode of transport's actually not very common for a virus. Often it is through an insect, through grafting, and also through pollen or the seed. So those ones that are through the natural reproduction of a plant are ones that we can really have a look at now, because they, of course, must be co-evolving with the plant. We've got examples here in New Zealand where we've got seed-transmitted viruses, but we don't yet understand the impact that virus is having on the plant. We're even selecting them for them in grafting, but that's another story. So we're at the very beginning of knowing the whole virome of the world. We're even less understanding the impact of those viruses. And yes, the proportion of pathogenic viruses is small, and that's really where we've focused to date.
Yep.
So there's this big, huge opportunity of understanding what they are and what they're doing.
Do you see then in this opportunity potential ways that we can deal with the challenges that we're facing today, like climate change?
Yeah, so bring us back to that example of the tolerance to heat or the tolerance to drought, and the ability, the characteristic that the virus can confer, at least in the short term, to some stress.
Right.
So those are examples of abiotic stresses, but there are also examples of biotic stresses, like the virus of the fungus. So I think that it's very timely that we have this high throughput sequencing, but it has to be married with the biology at a time when we're needing to find answers to how do we adapt ourselves and our crops our understanding of this beautiful earth that we're living on. How do we adapt to the climate change we've imposed on it, and perhaps even ways of imposing less climate change effectors? Can we use viruses, good viruses, instead of some of the chemical methods that we use to control our pathogens? So they have a lot of promise in how we can understand and take advantage of them.
Absolutely. So that sounds like the future of virology right there.
Yeah, it is. It's the future of virology. It's very exciting. You know, we've really come to understand that we as individuals, and not just individuals, actually human cells are a smaller proportion of us than our total. We're very much dependent on our microbes, and we understand the concept of good bacteria. And I really encourage the opening of the eyes, the opening of the mind, that there are good viruses. We've just been constrained by our technology, and yes, sometimes funding, granted. Oh, granted, that's funny, isn't it? That there are good viruses out there, and we need to discover them now. We have the tools to discover them. Let's find them and benefit from them.
Absolutely. It's been a fascinating talk with you today. Is there anything else you wanted to say about good viruses that we didn't quite cover?
Well, I think we've covered a lot. I think that the main message really is in the rap.
Okay.
Don't go giving viruses the bad rap. They have potential to do good things, and those good things are numerous. Let's get into virology. Let's understand how they're impacting our microbes, how they're impacting our insects, how they're impacting our plants, how they're impacting us, how they are master puppeteers of the ecosystem. If we follow the viruses, I think we can learn a lot about the ecosystem that they are actually controlling.
Fascinating. I'm absolutely with you. Thank you so much for your time and expertise today.
My pleasure, Hilary. Thanks very much.
And thank you out there for listening. If you'd like to know more about the SciGest episodes, please check out the Plant and Food website at plantandfood.co.nz, which has extra information about each SciGest podcast episode. Please also feel free to subscribe to the SciGest channel and rate and review each episode. Why not give it 5 stars? And thank you, and